Welcome back. This is your pre-lab for part 2 of the diffusion through a membrane state-mandated lab. So again, because it is mandated by the state, you have to have this lab in order to take your Regents exam in June. So it's super important that you guys complete this pre-lab as well as the lab tomorrow in class, okay? So before we get started, you guys have got 2 do not questions. These do not questions are previewing some things we're gonna do today in lab. So go ahead and pause this video, take an educated guess about those questions, and then press play when you're ready to review them. So question number 1 says, when preparing a wet mount of onion cells, which we'll do today or tomorrow in lab, a student put a drop of Lugol's iodine solution on the slide. Lugol's iodine solution was applied in order to 1, prevent air bubbles, 2, make cell structures more visible, 3, increase the magnification, or 4, increase respiration in the cells. So you guys may have had some trouble with this one, but let's break down what we know, okay? So we know that onion cells are going to be clear, right? But Lugol's iodine solution, we know is an amber color. Okay, so what ends up happening is when we put that amber solution on top of our clear onion cells, it's going to make our cell structures more visible. So the answer here was B, make cell structures more make cell structures more visible. And this is a technique called staining, where you're going to physically stain the color of the cells in order to make the things inside easier to see. All right, and number 2, the diagram to the right shows how a cover slip can be lowered onto some single-celled organisms during a preparation of a wet mount. Why is this a preferred procedure? Is it 1, the cover slip will prevent the slide from breaking? 2, the organisms will be more evenly distributed. 3, the possibility of breaking the cover slip is reduced. Or 4, the possibility of trapping air bubbles is reduced. So in this example, we see that we have our cover slip up top, and we have our slide down here at the bottom. And whenever we make a wet mount slide, you're going to drop that cover slip onto the slide at a 45-degree angle. And we do this in order to prevent the reduce the possibility of trapping air bubbles underneath that slide. So if you drop the cover slip directly on top of the slide, usually you'll get a giant air bubble in the center of your slide, which makes it really difficult to see the organisms underneath. So we have to drop it at a 45-degree angle to minimize those air bubbles that we see on the slide. All right. Moving on to our pre-lab. So the very first thing is we're going to review how to actually make a wet mount slide. Now, ideally, you guys would actually be doing this in the lab, but because we're all at home, we're having to kind of modify what we do. So if we were in the lab, we would use a dropper to place a drop of water onto the center of a slide. So you'd use this dropper right here. We would place a small, thin section of the sample on top of that drop of water. Now, this sample has to be super thin. And there's a couple of reasons we do this. First, it has to be thin enough for that cover slip to fit directly on top of the slide. If it's too big, that cover slip is going to fall off. And secondly, we want it to be a super thin section because we want to make sure we're just getting 1 layer of cells. Especially with today's onion cells, if we have too thick of a sample, what ends up happening is we have multiple layers of cells on top of each other, and it makes it pretty difficult to see what's inside. Okay, so we're going to put water and a thin section of our sample on that slide. And then we're going to place our cover slip on top, again, at an angle of 45 degrees to reduce air bubbles. All right. So on this PowerPoint, whatever I have written in red is what you guys need to fill in on your notes. So in this lab, we're going to be preparing a wet mount slide. And we will also need to label all of the organelles that we see. So we're going to be bringing back that Unit 2 knowledge. We're going to be labeling our organelles. Now in this lab, we're going to be using a red onion. Okay, and with that red onion, we need to make sure that we're taking a sample of the onion that contains red pigment in order to see what's inside the cell. So because of that, we're going to be using the outer layer of the red onion, which will give our cytoplasm inside kind of a purple or a reddish tint. And you guys will see what that looks like tomorrow in lab. All right, so before we get started, we need to review what we already know. So the first thing we need to review is whenever we're talking about the movement of molecules from an area of high concentration to low concentration without the use of energy, this is going to be called diffusion. So again, that's like going down the slide Oh, his leg's flying up in the air. Hold on. Whee. We're going from high to low, and no energy is required for this to happen. This is diffusion or passive transport. So we can see what that looks like in this picture. Up top, we have a high concentration of molecules, and down below we have a low concentration of molecules. So what those molecules do over time is they actually move across that lipid bilayer from high to low concentration until this very ending state when we have equal amounts or equal concentrations of molecules on both sides. This is what's known as equilibrium. But again, this is a review, so we should know this. That brings us to the first thing you guys need to do, okay? Now, I want you guys to either describe and or draw. I would prefer if you draw, but I know some of you guys don't have that capability. So draw a typical red onion cell mounted in water. So because it's a red onion cell, this is just a plant cell. So basically, draw me a normal plant cell. What does it look like? Think back to unit 2. What does a plant cell look like? So go ahead and draw me that cell and label the cell wall, cell membrane, and cytoplasm. Pause this video and press play when you're ready to review that. All right, so the cell you should have drawn me should look something like this. Maybe you drew some more organelles, maybe you just drew the bare minimum, either is fine. But you should have labeled 3 organelles. Your cell wall should be that rigid exterior of the cell. So it's that rigid outer layer or outer wall. Your cell membrane is the more flexible interior wall, or interior membrane, I should say. Okay, so the cell membrane is that flexible interior membrane, and the cytoplasm is that jelly-like center of the cell. Now, I do want to note one thing. Today in this lab, because we're dealing with a red onion, I told you guys that the inside of the cell is going to be like a purple color. So I'll draw you what that should look like on here. Okay, so tomorrow in lab, when we see this cell, it's going to look something like this, where the inside of that cell is going to be purple. And again, we do need to note that, remember, the cell membrane directly, oh no, let's do that in a different thing. The cell membrane directly surrounds the cytoplasm. All right, so this is roughly what your cell will look like tomorrow in lab. Now, we've got one more thing to review, and that is the movement of water. Okay, so whenever water moves from an area of high water potential to low water potential, we call this osmosis. And this is basically just the diffusion of water. So again, we're still moving from high to low. We're still not using any energy because this is still diffusion or passive transport. But when we're talking about osmosis, we're just talking about the movement of water. So no solute is moving, just water. Okay. Now, in the next part of this lab tomorrow, we're going to be taking that regular plant cell and we're going to be adding a salt water solution to it. So salt water is a solution containing both salt and water. Now, where there's a high concentration of salt, meaning there's a lot of salt, there's going to be a low concentration of water. Because of that, water is going to move towards the area that is high in salt in order to balance out the system or equalize those concentrations. So again, water is the only thing moving because this is osmosis. Okay, so now I want you guys to think, what's going to happen to our cell when we add a salt water solution? So go ahead and draw me a cell. Again, it's still a red onion cell, still a plant cell. Oh no, there you go. Still a plant cell, but predict what will happen when we add a salt solution to it. So I still want you to label the cell wall, the cell membrane, and the cytoplasm in your drawing. All right, so here is our cell that we started with. Again, we've got the cell wall. Did I label it here? Nope. The cell wall, the cell membrane, and the cytoplasm. And we're adding a salt water solution, meaning that there's going to be a high concentration of salt and by default, a low concentration of water. But in our cell, we know that the cytoplasm is made of mostly water. So because of that, there's a high concentration of water inside the cell. So because we're dealing with osmosis here and water is going to move across the membrane, water is going to move from this area of high concentration inside the cell to the area of low concentration outside of the cell. So water is actually going to leave the cell and flow outside. And what ends up happening when water leaves our cell is our cell's gonna look something like this, where it looks like the cell membrane and the cytoplasm actually shrink. So I'll go ahead and label that. So notice that our cell wall on the outside has not moved. Remember, it is a rigid structure. It does not move, but the cell membrane does. So the cell membrane and the cytoplasm appear to shrink. Okay. But again, because in the lab we're dealing with a red onion cell, the inside of the cell will look kind of purplish. So I'll go ahead and fill that in, show you guys what it will look like tomorrow. So the cell membrane and the cytoplasm shrink, and the inside of that cell will be a nice purple color. Oh, there's my labels. I knew I did it. Okay, so again, the cytoplasm and the cell membrane appear to shrink, but the cell wall remains the same. So what you guys need to write down is that when salt water was added to the onion cell, the cell membrane and the cytoplasm appeared to shrink in size. This is because water has left the cell through osmosis, since the salt solution or the salt concentration is higher outside of the cell. But it is important to note that the cell wall will stay the same. Remember, the cell wall is a rigid structure. Okay, but the cell membrane is flexible. All right, the next part of our lab, we just added salt water, and now we're just going to add regular distilled water solution. So this is just pure H2O. Okay, we're going to be adding distilled water back to that cell. But before I show you guys what's gonna happen, I want you to pause this video and draw me a picture of what you predict will happen. So when we have a red onion cell and we add pure water to it, what's it gonna look like? Okay, so make sure you label your cell wall, cell membrane, and cytoplasm. All right, so here is the cell that we're starting out with. Again, we just added salt water to it, so the cell membrane and the cytoplasm appear to have shrunk. But when we add distilled water or pure H2O, pure water to this cell, we're now going to have a high concentration of water outside the cell. And because all of the water has just left our cell, we're going to have a low concentration of water inside that cell. So because of that, again, it's osmosis. Water is going to be flowing from high to low concentration. Water is going to flow into our cell from high to low concentration. And what ends up happening is your cell is going to look something like this. So that cell membrane and the cytoplasm are going to expand again. But again, the cell wall does not move. So your cell wall remains the same, but your cell membrane and your cytoplasm appear to expand because water has just flown inside the cell. Okay, but one more time, I'm going to show you guys what that would look like if it were purple, like we'll see in tomorrow's lab. So I'll go ahead and color this in. All right, so there we go. Again, the cell wall does not change. Cell membrane and the cytoplasm expand. Right, so what happened here? So what we need to write down is that when distilled water was added to that cell, the cell membrane and the cytoplasm appeared to grow in size. They expanded. And this is because the distilled water entered the cell since the water concentration was higher outside of the cell. So again, with osmosis, we're going from high to low water concentration. So water flowed into the cell. All right, and that is all I have for you guys today for this pre-lab. Your last thing, remember to complete your exit ticket and turn this in before tomorrow morning at 8:30. All right, guys, have a wonderful day, and I will see you tomorrow.